Today we are going to discuss about the media composition, which media is applicable or which media we are using in case of plant biotechnology or the tissue culture technology. So as we know the media is having nutrients. These nutrients are important for growth and development of plants. These media are important for growth and development of plants. So media will be success. So depends matlab how we will be able to say that this is the complete media or this media is having nutrients or essential components which is helpful for the growth. First is the media which we are using, and the second is the media which are using for tissue culture should be able to start cell division. This is a crucial step, that when we are taking our explant onto the media, the first crucial step is that the explant should be able to start its cell division. When the explant starts its cell division, it means the nutrient which we are providing are having ability to initiate its cell division. When it starts its cell division, then obviously then the growth will occur. Then growth will occur. So we have to be able to establish media and maintain the media. We have another factor is the establishment of the media. So we have to establish the media for our tissue culture lab or for tissue cultures, and we should be able to formulate that media. We should be able to formulate that media in a proposition, in a equal concentration, or we should be able to know the what are the macro or the micronutrients which will be applicable for tissue cultures. As we know the plant medium, it will be obviously complex in nature. Different plant species are having different media requirements. Means like we won't be able to use one specific media for all plant species. As plant species varies, their nutritional requirements varies. Their nutritional requirements vary. So obviously the artificial media which we are providing them to grow these explant under in vitro condition will vary. So these are some important parameters which we have to take into consideration while formulating the media. So obviously when we start our practical, if we are starting practical with the known species, if it is known to everyone, obviously there will be the availability of media, or the composition of the media will be known to us. So it's easy to start with the known species. But when we are starting our when we are starting our experiment with the unknown species, means it means none of the scientists have worked on that species. That time we have to formulate that media. So it means we have to go for trial and error based techniques. Sometimes we may give media, or sometimes we may add some nutrients which are not utilized by the plants. That time we have to eradicate. So it means simply हमें simple एक test करना है, where we will be able to know the each and individual component of the media. So as we know that we are having macronutrients or major inorganic nutrients, or micro major inorganic nutrients is the one, or we can call this as a macronutrients. Then second is the micro inorganic nutrients, or we can call this as a micronutrients. The third one is the iron, which act as a chelating agent. Fourth is the vitamins. We have to add the vitamins into the media. Fifth is the carbon source. As in case of plant biotechnology, we mostly use sucrose as a carbon source. Why sucrose? This is cheaply available. This is inert one. And most important is when we autoclave our media, this sucrose is stable after autoclaving. So that is the reason we mostly use sucrose as a carbon source in the plant tissue culture. Then another are organic nitrogens. We have to add plant growth regulators, and at the last we have to add gelling agent. We have to add gelling agent, that is our agar. Agar is used, simple agar. Agar is not that costly as agar roses which we are using in the molecular tech biotechnology, molecular biology. So if we'll talk about the media, since 1902 we have started preparation of media, but Murashik and Skoog, these were the two scientists who first developed the artificial media in the year 1962. Murashik and Skoog. We called their media as a MS media. We call this is one of the common media which is applicable to the most of the plants, because this media is having higher concentration of salts in it and plus higher concentration of sucrose. We will see this later on. So when Morashik and Skoog were working, they were working on the tobacco plant, for which they analyzed the mineral components present in the plant. They analyzed the mineral components present in the plant. As per that analysis, they were able to make MS media. Okay, and in this, we are, as I said, they are having relatively higher salt concentration as compared to the other medias which are commercially available nowadays. That media are used for the tissue culture technology. There are two minerals which are mostly which are present in the higher concentration. One is the potassium and another is the nitrogen. सिंपल अगर ये बोला जाएगा, एमएस मीडिया जो है वो बनाया गया था किसके लिए? एनालिसिस कर मिनरल्स का एनालिसिस करते हुए टबैको प्लांट से, मतलब उन्होंने टबैको प्लांट लिया जिसका उन लोगों ने मिनरल एनालिसिस किया. तो मुराशिक स्कूल का एक और पॉइंट ये है कि दे आर हैविंग हाई सॉल्ट कॉन्सेंट्रेशन, particularly potassium and nitrogen. This was about the MS media. Then another is the Linzmeyer Skoog media. Another is the Linzmeyer Skoog media. They developed this media in 1965. Murashik Skoog had developed this in 1962. Phil Linzmeyer Skoog developed another media which is the replica of actually MS media, but they have modified the organic constituent of the media. They have modified the organic constituent of the media. They developed this media in 1965. Then another group of scientists came. That media they made in 1963. They called this media as White's media. White was the scientist who made this media. So this media is named as per his name, White's media in 1963. This media is formulated with the low salt concentration. This media is formulated with the low salt concentration. सिंपल टर्म्स में याद रखें कि एमएस मीडिया में हाई सॉल्ट कंसंट्रेशन होता है और जो हमारा वाइट्स मीडिया उसमें लो सॉल्ट कंसंट्रेशन होता है. और जो हमारा लिंसमेयर स्कूग मीडिया है, दैट इज़ द वर्जन ऑफ एमएस मीडिया जहाँ पे थोड़ी सी सॉल्ट कंसंट्रेशन कम है एज कम्पेयर टू द एमएस मीडिया. These were the three medias. Then last but not the least, Gambox, Gamborgs. Gambox B5 media is the another media. This was developed by Gamborg et al. Means बहुत सारे साइंटिस्ट ने इस पे काम किए in the year 1968. This Gambox B5 media is also called as simple B5 media. When these scientists were working on soybean callus culture, so that time they were able to develop this media. In this media there is a greater proportion of nitrate. B5 media में क्या आसान ज़्यादा nitrate as compared to the ammonia. Now if we will talk about the salts which we have to add as a macronutrients, so obviously as name indicates, macro means these we have to add these nutrients in the media in large quantities. The quantity are measured in millimolar per liter. mm, small m, capital M, then per liter, millimolar per liter. And these macronutrients include nitrogen, phosphorus, potassium, sulfur, magnesium, and calcium. Again, I will repeat: nitrogen, phosphorus, potassium, sulfur, magnesium, and calcium. All these macrosalts or the macronutrients are involved in the structure and functional process of the plants. All these macronutrients are involved in the structure and functional process of the plants. As if we will talk about in the protein synthesis, we know till now in protein synthesis we are having mostly involvement of nitrogen and sulfur is there. Or if we talk about the nucleotide synthesis, so we are having phosphorus there, nitrogen there, sulfur. Or calcium is utilized in the cell wall synthesis. Or magnesium act as a coenzyme or cofactor. And for the membrane integrity we need magnesium ions. We need magnesium ions. So this was about the macronutrients which are present in the plant tissue culture. If we will talk about the micronutrients, we are having boron, manganese, zinc, molybdenum, copper, and cobalt. We are having boron, manganese, zinc, molybdenum, copper, and cobalt. As the name indicates, we have to use these nutrients in a very minute quantity, micromolar. Micromolar per liter, we have to add this. But there are some even like macronutrients where we were not able to see their role in the plant tissue culture. Means हम जब हम लोग experiment करते हैं we have to add them, but specifically we don't know what is their exact role. There are some macronutrients micronutrients available in the medium for which we don't know the exact role where they are involved in the tissue culture growth and development. Like if we talk about the zinc in tissue culture, we provide this zinc in the form of zinc sulfate. This zinc neither get oxidized nor get reduced, but this zinc act as a metal component to many coenzymes. ये जो सिंपली एक्ट करें क्या जिंक मेटल कंपोनेंट बहुत सारे एंजाइम्स को ये एक्टिवेट करता है. Then it has been seen that the zinc is involved have active role or involved in the protein synthesis, and most importantly it has been observed that this zinc play an important role in the synthesis of tryptophan amino acid. Means zinc ज़्यादा किसके लिए important है? Protein synthesis, and most importantly it's important in the tryptophan. This was about the zinc. If we will talk about the copper, this copper has role in the photosynthesis. This copper plays an intermediate role in the electron transport chain when the electrons are transferring from photosystem 1 to photosystem 2. It has been observed that when we are giving minimum quantity or less amount of copper to the plant, then there is the decrease in the rate of photosynthesis. Then there is a decrease in the rate of photosynthesis. If photosynthesis is not occurring, they are not able to make their food, then there will be stunted growth. And this copper is provided to plants in the form of copper sulfate. This is copper is provided to plant tissue culture in the form of copper sulfate. So next is our iron, which third one is our iron, which act as a chelating agent. So this iron is important for it has been seen the role of iron in the coenzymes. They are also, or we can say, cofactors. It has been seen that the availability of iron in a free state is in the plant is very low. Means iron free state में बहुत कम पाया जाता plants में. It's 1 to 10 millimolar. And it has been seen that if there is a deficiency of iron to the plants, then there is stunted or severe effect on growth and development of plant cells. क्योंकि एंजाइम्स काम नहीं करेंगे तो रिएक्शन प्रोसेस नहीं होगी ज़्यादा, तो उसकी वजह से क्या होगा, अल्टीमेटली जो हमारी प्लांट की ग्रोथ है दैट विल affect, and obviously the development of the plant will affect. Now another is the vitamins. Now another is the vitamins. Plants are able, or tissue culture cells are able to form their vitamins, but they are able to synthesize in a very low quantity. The need of plant is much more than what they synthesize. So in other words, we can say that they produce these at suboptimal level. So exogenously or from outside, we have to supply the vitamin source in the tissue culture to the plant source. And the most commonly which we are adding vitamins are thiamine, vitamin B1, pyridoxine, and nicotinic acid. There are some natural components, we don't know exactly their components, but these natural occurring components are added to plant as a supplement, because we don't know, like if we talk about the coconut water, it's undefined substance, we don't know the complete composition of these substances, but it has been seen that it's not exactly essential, but that components which will be present in the coconut water will enhance plant cell growth and development. So like we are having many natural complexes, like we use endosperm fluid or coconut milk. So this endosperm fluid or the coconut milk is taken from the coconut fruit. Sometimes we use many juices like fruit materials we take. These juices are taken from either orange or tomato. Sometimes even we are using potato extract. So this potato extract has been extracted from the potatoes itself. So extracts of matlab yeast and malt, we extract yeast and malt from the malt and yeast. माल्ट मतलब होता है जो बार्ली को जब हम लोग क्या करते हैं, यू हैव read this in beer fermentation, so you are familiar with what is malt, right? And there are some animal extract, like um, in case of animal biotechnology, we use animal extract like blood, we use there as a supplement. But for plants, we use mostly fish emulsions. We use fish emulsions as a supplement for plant growth and development. Now another factor which are present in the media are plant growth regulators. Plant growth regulators, or simply one of plant hormones. These are actually the organic substances which have physiological role, and these plant growth regulators are needed to the plant in a very low concentration, less than 1 millimolar, or even sometime we can say less than 1 micromolar, means इतना छोटा quantity उसकी ज़रूरत होती है plant को. So if we add these phytohormones to the plants, they change their phenotypic characters. As we know, if we will provide oxygen to the explant, there is a formation of shoot, and this is a phenotypic character. As we are able to see this formation of root, or if we will take the higher concentration of cytokinin to oxygen, there is the formation of shoot formation. So it matlab like plants will be able to produce it naturally or synthetically. We have to add these plant growth regulators to the medium. So this was the basic intro for the media. So next part is we have like different medias with different concentrations. First we will take our example with the White's medium. First is the White's medium. This White's medium is mostly developed for root culture. This medium why we proceed for root culture, there are some plant roots which are able to produce significant importantly secondary hormones. So for that reason we are our secondary metabolites, sorry, secondary metabolites, we are able to extract secondary metabolites from some roots. So there we are using this whites medium. So the macronutrients which are present in the whites medium include MgSO4 with the concentration of 750 milligram per liter. Again, I will repeat, magnesium sulfate with the 750 milligram per liter. Then the second component is NaH2PO4. This is the composition, and the amount utilized for the white medium is 19 milligram per liter. Now the third macronutrient is KNO3. This we are adding to whites medium 80 milligram per liter. So the another component is the micronutrient. This is H3BO3 with the amount of, or with the concentration of 1.5 milligram per liter. Another is MnSO4 5 milligram per liter, zinc sulfate 3 milligram per liter, CuSO4 0.01%, sorry 0.01 milligram per liter, potassium iodide Ki is 0.75, and here we are using sucrose 20 milligram per liter of the medium. And the organic supplements or the vitamins which are added to whites medium include thiamine 0.01 milligram per liter, pyridoxine 0.01 milligram per liter, and nicotinic acid. Their concentration is 0.05 milligram per liter. And glycine we have to add 3 milligram per liter. So this was the complete composition of the whites medium. So this white medium is used with the pH 5.8. This medium is used with the pH 5.8. Now another is our important media which is mostly applicable in day-to-day plant tissue culture labs, that is the Murashik and School medium, MS medium. So this Murashik and School medium are having macronutrients like MgSO4, KH2PO4, KNO3, NH4NO3, and CaCl2. So their amount is like for MgSO4 we are using 370 milligram per liter, KH2PO4 in the concentration of 170 milligram per liter, KNO3 with the concentration of 1900 milligram per liter, KH4NO3 with 1650 milligram per liter, and calcium chloride CaCl2 with 440 milligram per liter. And for Murashik and School, micronutrients include H3BO3 with concentration of 6.2, MnSO4 22.3. All the concentrations are in milligram per liter only. ZnSO4 8.6, Na2MOO4 0.25, CuSO4 0.025, COCl2 0.025, Ki potassium iodide 0.83, FeSO4 27.8, sodium EDTA, Na2 EDTA 37.3. And here we are using sucrose. This sucrose is having higher concentration like 30 milligram per liter. 30 milligram per liter. And vitamins we add thiamine 0.5, pyridoxine 0.5, nicotinic acid 0.5, and myoinositol 100 milligram per liter. And again we have to add glycine 2 milligram per liter, and we are keeping its pH at 5.8. As we can see the list, like what we discussed, that they are having higher salt concentration as compared to the B5 medium, and they are having 30 milligram per liter of sucrose, while in case of whites medium we use it 20 milligram per liter. And the last one medium which we are discussing is the B5 medium. This B5 medium is mostly designed for cell suspension cultures and callus cultures. When we have to proceed for cell suspension culture or callus culture, that time we are using this B5 medium. The macronutrients include MgSO4 in the concentration of 250 mg, 250 milligram per liter, NaH2PO4 150, KNO3 2500, CaCl2 150, ammonium sulfate 134. Macronutrients will include H3BO3 3 milligram per liter, MnSO4 10, ZnSO4 2, Ne2MO4 0.25, CuSO4 0.25, and cobalt chloride COCl2 is 0.25, potassium iodide 0.75, and for sucrose we are using 20 milligram. In case of ms media we are using 30. For rest of the medias we are using sucrose 20 milligram per liter. And the vitamins are thiamine 10 milligram per liter, pyridoxine 1 milligram per liter, nicotinic acid 1 milligram, minoxidil 100 milligram, and we have to keep this pH at 5.5. This is the how the media are formulated. And but most importantly composition is important, and another is the factor is the pH. Another factor is the pH. So pH also play important role in the development of plants.